Hello students, today we are going to start with a new topic, excretion and osmoregulation. In this, we are going to learn about the modes of excretion like Ammonotelism, Ureotelism, Uricotelism, also Guanotelism. Other than that, plasma creatinine. Also we learn about osmoregulation, osmoconformers and Nephridia, which is a excretory organ in the lower organism or in case of non-chordates, and few more other organs related with the invertebrates. Also in the present slide you can see human excretory system. Now as we know, human excretory organ is the kidney, and you can see other parts, ureter, urinary bladder and urethra, right, in humans as well as the mammals. So in today's part we are going to learn about the modes of excretion and other content which is present along with the organs in invertebrates. Okay, now the introduction. Array of chemical processes occur in the body of an organism. Sum total of these processes is called as metabolism. So all the processes, or to be very precise, chemical processes which takes place in the body is called as metabolism. So metabolism involves catabolic and anabolic process. What is catabolic? Breaking down, whereas anabolic, building up. So metabolism produces a variety of byproducts, some of which need to be eliminated, otherwise it can cause harm to the body. Such byproducts are called as metabolic waste. Metabolic waste products might be fluid, that is liquid, or it can be gaseous, or organic or inorganic component. Depending on the type, they are eliminated through various organs of the body of an organism. So in general, as we saw in the first slide, kidney is the main organ in case of humans and mammals. In other animals, there is nephridia, or in mammals itself, there is skin or lungs, which also eliminate various components from the body. Now in the present slide you can see two images. One is metabolism, where you can see the chemical energy of carbohydrates, fats, proteins, others, and chemical waste, carbon dioxide, water, and where ATP is formed along with heat is released. The ATP, which is the body energy currency, further breakdown takes place along with heat is released. Now you can see the second image on the right hand side, where simple molecules such as glucose, amino acids, glycerol and fatty acids, which are formed with a catabolic reaction. Further, these anabolic reaction transfer of energy from ATP molecule to complex molecule. Again heat is released, and further the complex molecules such as starch, proteins and lipids are formed. Again these are broken down by catabolic reaction, transfer of energy from complex molecule to ATP, and again it is broken down by releasing heat and formation of simple process. So as you can see, catabolic, the complex substances are broken down, anabolic, the formation of simpler components taking place. And also towards the central part of the image we can see ATP formation when catabolic reaction takes place, and also whenever the energy is required, the ATP is broken into ADP and IP, that is inorganic phosphate, and again it combines to form ATP. Now we come into the detailed part of excretion and excretory products. First point, elimination of metabolic waste product from the body is called excretion. Now when you say metabolic waste product, or we can use a precise term again, nitrogenous waste product, right? Always there's a small confusion. Elimination of waste product is also called as egestion, right? So egestion is only elimination of waste product, but when it comes to metabolic waste or nitrogenous waste, it is excretion. Further, unlike digestive waste, which are primarily composed of unabsorbed or undigested substances that have never entered the cells, metabolic waste are produced inside the body cells. That is the food which we take in, which is broken down, and the metabolic waste is produced, as you saw in the previous slide image. Various excretory waste products produced in the human body, that is fluids such as water, gaseous waste like carbon dioxide, nitrogenous waste like ammonia, urea and uric acid, along with creatinine, minerals, Also salts of sodium, potassium, calcium, etc., which if are excreted through urine, feces and sweat, as this has to be removed out of the body, because if it is present in the body, it is stored in the body, it can cause various disease and disorders. Further, pigments formed due to breakdown of hemoglobin are bilirubin, which is excreted through the feces, and urochrome eliminated through the urine, which also give the coloration to feces and the urine. Further, also the pigments present in foodstuff like beetroot, which you can see blood red color, right, consumed by organism, or excess of vitamins, hormones and drugs taken are also eliminated through the body. If these are not eliminated, it can cause harm. And how these are present in our body, as we just read, if you are taking foodstuff which has got lot of pigments present in it, or if you are taking in vitamins, calcium tablets, or any medicines, drugs for any health issues, all these are eliminated through the body. Further, spices we consume contain volatile substance that are excreted through lungs. As we know, spices increase the taste of the food, but if it is present in more, it is again not good for the health. But these are volatile substances which are excreted through the lungs. Body of an organism can store excess carbohydrate and fats, but is unable to store excess amino acid. That is, as we know, fats and carbohydrate can be stored in the body, but it may lead to obesity or various other problems, but it can be stored. But in case of amino acids, it cannot be stored in excess. Further, hence excess amino acids are essentially broken down by a process called as deamination. In this process, amino group is separated from the amino acid and ammonia is formed. So this statement is very clear how ammonia is formed by deamination process, that is breaking down the amino acids. Further, toxic ammonia is either excreted as it is or further converted to less toxic forms like urea or uric acid before excretion. There is no clear correlation between the phylogenetic relationship of organism and the major excretory products, that is, its habitat has. Now here when you mention phylogenetic relationship, that is the evolutionary relationship, so in that doesn't give a clear relationship between the excretory products and the species, because if you see from species to species or within the species, there's a differences, but the species where it is present, the habitat, surrounding environment, accordingly they secrete the excretory product. Like tadpole of a frog excretes ammonia as it is present in water, and adult frogs mostly ureotelic because they are present in water but also on land. Further, some terrestrial turtles excrete uric acid whereas others excrete ammonia. So you can see in terrestrial turtles itself there's a differences. Certain produce uric acid and certain urea or ammonia. Animals can be broadly classified into three types based on nitrogenous waste, as we read in the first part, that is ammonia, which is a basic product of deamination process, but it is highly toxic. Hence, it is to be diluted immediately. So, ammonia requires lot of water. If there is no or limited access to water, need for conversion of ammonia becomes necessary. So, that is, if the species present a habitat where water is present, they can eliminate ammonia. If water is not available, then they have to immediately convert that ammonia into either urea or uric acid. Otherwise, it will be harmful for the species. Thus, availability of water plays key role in deciding mode of excretion of an organism. So, water plays an important role. These are mainly three types, that is ammonotelism, the one which secretes or not secretes, excrete. Here also the term difference, the secretion, excretion. So, it is excretion. Okay. Ureotelism, the one which excretes urea, and Uricotelism, the one which excretes uric acid. Clear? Again here you can see an image. Excretion, there is disposal of nitrogenous waste. Ammonia, too toxic to be stored. Land animals must dispose of nitrogen in the form of urea or uric acid, because land animals water availability is less. Further, urea can be stored in concentrated form but requires water for disposal. So urea can be stored but it has to be removed with the help of water. Uric acid allows for water conservation. So again you can see the image, proteins which are broken down into amino acid, nucleic acid into nitrogenous bases. Further, they both form amino groups. In case of aquatic species including fishes, it is ammonia as water availability is in plenty. Mammals, amphibians, sharks, some bony fishes, which is urea, and birds, insects, many reptiles, land snails, all uric acid. So in the further slides I have shown a lot of animation images related to species which excrete ammonia, urea and uric acid, so which will help us to understand very well about the excretion and modes of excretion. Further, now we go into the individual parts. First one, ammonotelism. So, elimination of nitrogenous waste in the form of ammonia is called ammonotelism. So, very clear to understand. The species which excretes out nitrogenous waste in the form of ammonia, as it is basic in nature, hence it would disturb pH of the body if retained. Slight increase in pH would disturb all the enzyme catalyzed reaction in the body and will also make the plasma membrane unstable. It is readily soluble in water and needs large quantity of water to dilute and reduce the toxicity. That's why ammonia or species which secretes ammonia requires water. So usually these species are present in water bodies or near to water bodies. However, it is energy saving mechanism of excretion. Hence, all animals that have plenty of water available for dilution of ammonia excrete nitrogenous waste in the form of ammonia. Such animals are called as ammonotelic. Now, when the first point what we learned, ammonotelism, that is the process of elimination. Here, the water availability, the animals which excrete, they are called as ammonotelic. So further, 1 gram of ammonia needs 300 to 500 ml, that is almost close to half liter of water is required for elimination of ammonia, just 1 gram, right? So it is so that's why the species are more present in water. Ammonotelism is found in aquatic invertebrates, bony fish and aquatic or larval amphibians. Animals without excretory system are also ammonotelic. Example, protozoans. Ammonotelic animals excrete ammonia through general body surface such as skin, gills and kidney. So towards the right side again I put image to understand the process little bit much more easier. The process of excreting ammonia, ammonotelism, as you have seen the definition. Further, many bony fishes, aquatic amphibians and aquatic insects are ammonotelic, or to be very precise, the aquatic species, mainly the freshwater species, they are ammonotelic. Further, osmotic balance, freshwater fish, how it is maintained, just to give an understanding. Food, fresh water, gills, active absorption of NaCl, water enters osmotically, intestine waste, that is urine, kidney excretion, dilute urine, and you can see the large glomerulus, NaCl which is excreted out by the kidney tubule, right? Active tubular reabsorption of NaCl. So here reabsorption also takes place. Now you can see beautiful images, also some unique species again. So I put species which are bony fishes, also something different. So you can see the first image towards the left top, bony fishes, you can see different species of bony fishes. Okay, to understand the species present in freshwater, we can say very clearly. Okay, so you can see the image continuously changing. Then second one from the top, tadpole larva. As you mentioned, tadpole larva, they secrete ammonia, whereas the amphibious one, urea, which is present on the land. Then one amazing species, mola, which is heaviest known bony fish. So it's like a unique species, little bit of additional information also. You can see a person swimming around the heaviest bony fish the nearby. So you can see the difference in the fish, right? The size you can make an judgment over how much heavy it will be. Then lower part towards the right, oar fish. See how the fish appears, very thin, long, which is appearing like a oar, which is used by the boatman. Hence the name given as oar, like the appearance. And towards the left, carp fish, which is again quite common. Observe different types, we can observe carp fish, right? Now coming to the second type, it is Ureotelism. Now first point, similar to the Ammonotelism. What is the first point? Elimination of nitrogenous waste in the form of urea. Then it is called as Ureotelism. Urea is less toxic and less water soluble than ammonia. So we can have a difference also over here. Hence it can be concentrated to some extent in the body. Means it can be stored for a certain period of time. Due to this, it regulates less water for elimination compared to ammonia, about 100 times less water in human, several hundred times in camel, kangaroo rat and shark. So we can see the difference. As it is less toxic and less water soluble, hence Ureotelism is suitable for animals. Those need conservation of water to some extent. Hence it is common in terrestrial animals, or we can say the land animals, as they have to conserve water. It takes about 50 ml of water for removal of 1 gram of ammonia in the form of urea. What is the difference in ammonia now? Ammonia requires almost 500 ml. So that's why it's mentioned. So we can see a huge difference in the excretion of 1 gram of ammonia, where 500 ml is required almost, here just 50 ml. Mammals, cartilaginous fish, sharks and rays, many aquatic reptiles, most of the adult amphibians are ureotelic. Now these, some of the species we can see in the next slide. They convert ammonia to urea in the liver by operating ornithine or urea cycle, or Krebs and Hansard in 1932. So it is commonly called the conversion like ornithine cycle or urea cycle. Further, 3 ATP molecules are used to produce 1 molecule of urea. Sharks retain more urea in their body fluid to make their blood isotonic to the surrounding environment, that is marine water. As you know, these are marine fishes and the water is very salty, so to maintain that they has to retain urea. This helps them to prevent possible loss of water by exosmosis. So again you can see the image like ammonia as I kept. Ureotelism, many mammals, many terrestrial amphibians and marine fishes mainly excrete urea as the urea and are called ureotelic animals. And again I put as image of a marine fish, where you can see food which is and seawater which is enters inside the mouth, gills, active secretion of NaCl, water loss, intestinal waste, MgSO4 voided with fat feces, kidney excretion of MgSO4, urea, little water. Further again you can see glomerular reduced or absent, so MgSO4 where active tubular secretion of MgSO4 takes place. Now you can see again here beautiful images or some unique ones. Again see the left one, Macropina microstoma. See again a unique species deep inside the water. It's a deep sea fish with transparent head. You can see the head is transparent and a tubular eye. Now one, if you see carefully, 2 eye spots are there, but actually it is not the eye. That's why we get confused. If you just see upper part, there's a tube-like structure above the eye spot, which is the actual eye. Okay, and then the middle, glass frog. We can see the transparent digestive system, the mouth, even the heartbeat. Now you can see as it zooms, you can see the now very clearly the heartbeat also. That is glass frog. We can see through into the intestinal part. On the right side, again you can see a lot of terrestrial animals. The slide is keep moving, so you can see the various species on land or terrestrial. Then lower right end, tiger shark, and then left, turtle. Now third type, Uricotelism. Elimination of nitrogenous waste in the form of uric acid is called Uricotelism. So now we can understand Ammonotelism, Ureotelism, Uricotelism, right? Further, uric acid is least toxic in comparison to urea and ammonia. Hence, it can be retained in the body for some time in concentrated form. It is least soluble in water. Hence, minimum, that is about 5 to 10 ml for 1 gram, or no need of water fluids elimination. You can see 5 to 10 ml is like few drops, we can say, correct? So the difference over here, 5 to 10 ml, which is for uric acid, in comparison to ureotelism or ammonotelism. See the difference, 5 to 10 in uricotelism, 50 ml in ureotelism, whereas 500 ml in ammonotelism. Hence, animals those need to conserve water follow uricotelism. So those needed to conserve, they follow this particular process, that is uricotelism. Ammonia is converted into uric acid by ionosinic acid pathway in the liver of the birds. Birds, sometime, or some insects, many reptiles, land snails are uricotelic because they need to conserve the water. Again, you can see the images in the next page, that is animation part. Here you can see quite a lot of examples in the right side image. However, they have to spend more energy. So here uricotelism, reptiles, birds and snails and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called as uricotelic animals. You can see various examples of uricotelic species. Now we can see beautiful again animations of uricotelic species. Right from left, giant African snail, very huge. The right one, snake, but here I put not the normal snake which all of you see the normal snake, right? That's why I always keep amazing species, unique species as an example to learn and to see also, which we come across very few times. So here you can see double-headed albino snake, right, very rare. Then right side you can see lizards. We have seen the house lizard, the other lizard, chameleon or calatus, etc. Right, here you can see another lizard, tree lizard, which is crested anole. We can see how fan like process spreading out in the neck region. Then in the middle, another we can say amazing species of butterfly, okay, which is green dragon butterfly, which is very unusual. We never come across such type of butterfly species. Then towards the left, birds, which is as we know it is a uricotelic species, but you can see various colorful species again animating different species, the common parrot, owl, but other than those we can see few more amazing colorful species. Now, moving further, now we can have a comparison. As while explaining, I was explaining in comparison to three, but here we can see all the three. So we can see a quick revision also, and the three types, differentiating types. Okay. Now, ammonotelism, nitrogenous waste in the form of ammonia. Ureotelism, nitrogenous waste in the form of urea. Uricotelism, nitrogenous waste in the form of uric acid, formed in the liver by deamination of amino acid. Formed in the liver by ornithine cycle, formed in the liver by ionosinic pathway. All the three pathways given, right? Then ammonotelism, least expenditure of energy. Ureotelism, relatively more expenditure of energy. Uricotelism, high expenditure of energy. Highly toxic compared to less toxic. Uric acid is non-toxic. Highly soluble in water. A good solubility in water is insoluble in water. Excreted rapidly in urine, excreted at a slower rate, excreted very slowly. Dilute urine, concentrated urine, thick paste or semi-solid pellets. And last point, 500 ml of water required for elimination of 1 gram of ammonia, 50 ml required for elimination of 1 gram of urea, and 10 ml of water required for elimination of 1 gram of uric acid. Now, coming to the other modes of excretion other than ammonia, urea, uric acid, that is guanotelism. So here we can see animals like spiders, scorpion and penguins, they excrete guanine. As we see, very few species. This mode of excretion is called as guanotelism, and I put an image first. You can go through the points again little bit, which makes it more interesting. Aquatic species excrete free ammonia through gills. Terrestrial Critters produce urea, very soluble, still needs water for removal by kidney, imposes a minimum daily water requirement. Spiders excrete guanine, 5 nitrogen atoms in small intestine. So you can see spiders in the center, and also scorpion, another image which I put, and towards the right you can see again beautiful image or animation of 3 species which excretes guanine or guanotelism, right? Jumping spider, spiders you have seen, but again jumping spider, and right side scorpion. Now it's not just scorpion, I placed one beautiful image where scorpion is carrying its babies on the back. If you see, there may be around 20 to 30 species on the back of the mother scorpion, right? Then lower the penguin, which you are all very familiar. Now, plasma creatinine. What is that? We'll go into the detailed part of it. Plasma creatinine is produced from catabolism of creatinine phosphate during skeletal muscle contraction. It provides ready source of high energy phosphate. Further, normally blood creatinine levels remain steady because the rate of production matches its excretion in the urine. So usually it is steady. Hence, level above the normal is indication of poor renal function. So whenever the level goes beyond the normal level, then we can indicate that kidney functioning has been affected. That is mainly in case of diabetic condition, etc. Further, excretory organs plays an important role in maintenance of constant internal environment of the body called homeostasis. It requires osmoregulation, the process of controlling solute concentration and water balance. It can be rightly said that composition of blood and internal environment is determined not by what mouth ingests but by what excretory organs retain. So we can understand this term very clearly. Not what we take in, but what the excretory organ retain in the body. Marine birds like albatross spend their life on the sea. That means water they drink is salty. How do they manage osmoregulation then? If they are taking in lot of salt water, they got special glands called salt glands near nostrils. These are capable of secreting salt by active transport and help to manage osmotic balance. Because of the salt glands, they can manage or are capable of secreting this excessive salt from the body. Many marine organisms like sea turtle and marine iguana also has got such salt excreting glands. Animals can either be isoosmotic to the surrounding, that is called as osmoconformers. Now we come across few new terminologies, or control internal environment independent of external environment, which is osmoregulation. So two terms, one is osmoconformers and osmoregulators. Marine organisms mostly are osmoconformers because their body fluid and external environment are isoosmotic in nature. Freshwater forms and terrestrial forms are osmoregulators. Whether conformers or regulators, most organisms can tolerate only a narrow range of salt concentration. Such organisms are called as stenohaline organism. Another term, stenohaline organism. What is those organism which has got a narrow can tolerate narrow range of salt concentration? Further, those who are capable of handling wide range in salinity are called as ureohaline organisms. So one which can handle, they are ureohaline, otherwise stenohaline. Example, barnacles, clams, etc. Unicellular forms has got contractile vacuoles which collect and discharge waste products outside the cell. Excretion in sponges takes place by diffusion of waste material in water, which is discharged through osculum. Now in case of hydra or sponges, it removes out through osculum, whereas in case of unicellular organisms, as you can see in the image, paramecium, amoeba, etc., where contractile vacuoles are present, which helps in the discharge or removal of waste product. True organs of excretion are found in those animals that show bilateral symmetry. So as we are learned in the chapter Kingdom Animalia, although higher animals, organism from Annelida onwards, they are all having bilateral symmetry. So their organs are well developed and true organs for excretion. Most common type is simple or branching tube that opens to exterior through pores called as nephridia pores, which is, as I told, in case of annelida, earthworm, etc., which again we will be seeing in the later part of the video. Now here just two images I have put. As I mentioned to the right, paramecium, amoeba and euglena. Right, they have got contractile vacuole present in all these three. Whereas towards the left, what happens when the level of creatine is too high? Just as I mentioned, what are the symptoms? Shortness of breath, nausea, vomiting, dry skin, headache, loss of appetite, and weight loss. So creatine is transported through bloodstream to the kidney. That's why we have to be careful. If creatine is high, one of the indication is diabetic condition and other symptoms as mentioned over here. Now we come across two terms, osmoregulator versus osmoconformer. So I just added few image over here to make it more easier. What is osmoconformer? Isoosmotic to surrounding marine organism must have a stable composition, right? So they are all what? Osmoconformer. Osmoregulator, not isoosmotic to the environment, so control internal osmolarity, enables animals to live in varied environments, land, freshwater, cost, energy. Now two other terms, cyanohaline, urohaline. What is cyanohaline? Animals that cannot tolerate sharp change in external osmolarity, both osmoconformers and osmoregulator. Whereas what is ureoline? Animals that can tolerate sharp external changes in the osmolarity, as we learned the example barnacles, right? And right side I had given the image for both osmoregulators, regulation in case of fresh water, and regulation in salt water, how in both species it is carried out. Now, other than kidney, which you are going to learn in the next part of the video, here you can see the few other major types of nephridia. One is protonephridia. These are network of dead end tubes called flame cells. These are found mostly in animals that lack true body cavity, example platyhelminthes. So which we can see the image also. Protonephridia also found in rotifers, some annelids and amphioxus. So in the left, first you can see the image of planaria, which is an example of platyhelminthes. You can see how the actual structure is: eye spot, cerebral ganglia, longitudinal nerve cords, transverse nerve, intestine, pharynx, mouth and anus. Mouth and anus, see where it is, at the posterior end, which is common, mouth and anus, and excretory canal. And you can see the excretory pore separately highlighted on the lower part. Excretory pore, flame cell, nucleus, cilia, tube cell. Now on to the right you can see the animation image of planaria, and see here now. In the first we can see the anterior end, that is the eye spot. Now we can see the mouth and the anus part tube, which is seen as the planaria moves. You can now see very clearly. Okay. Now, second, from protonephridia to metanephridia. These are unbranched coiled tubes that connect to body cavity through funnel-like structure called as nephrostome. Body fluid enters the nephridium through nephrostome and gets discharged through nephridiopore, that is example earthworm. As mentioned, annelida has got nephridiopore, nephridia, which helps in the excretion. In most of the insects, excretion takes place by a set of blind-ended tubules called as Malpighian tubules. Now, when you go higher order in invertebrates, after annelida comes phylum Arthropoda. In Arthropoda, the insects, the very best example, as you can see, ant or cockroach, they have got Malpighian tubules. Whereas other arthropod like a crustaceans, they have got green gland as excretory organ. Members of phylum Echinoderma, much higher, do not have any specialized excretory organ. Waste materials directly diffuse into water or excretory through the tube feed. This also in the further slide you can see the tube feed, and also we can see how the excretory part, how it is expelled. Mammalian kidneys are a collection of functional unit called as nephrons. Again, I have kept an image to the later part of the video, and which we learn in detail in the next part of the video. which are well designed to extract metabolic waste. In both earthworm left, you can see the nephridia, all the parts are there, but you can see very clearly the nephridium, which is present towards the right side, where clitellum, septum, muscle layers, nephridium is present, and other parts of the body. And in the right side, you can see the ant, the internal digestive system, where the Malpighian tubules are present in the midgut region uh, in all the insects. Now here again you can see image of crayfish. Okay, now that is crustacean. So you can kept a animation image of crustacean in the lower part, and above you can see the image showing all the labeled part of the species, so that you can understand the part where it is present, what are the different labeling. If you just go through quickly, eye, below it the green gland is present, then mouth, mandible, esophagus, maxilliped, ventral nerve cord, ganglion of ventral nerve cord, digestive gland, flexor muscle, anus. At the top end, encephalon, stomach, heart, gonad, exterior, extensor muscle. So you can see green gland just lower towards the eye region, and towards the right the starfish, where again if you see the image picture which is there, you can see tube feet, and then the small tubes highlighted over there. Now I kept a image animation where you can see the live starfish, how the part of the tube feet which is seen, and the from the tube feet the radial nerves, how it is emerging out or moving around, right? So both the images help us to understand about it, and specifically the tube feet, how it functions. Now the mammalian kidney, which we will be learning in detail in the next video, that is the human excretory organ. So just for an idea, you can see the two animation images are there, two images pictures are there, and towards the center you can see the mammalian kidney, calyces, renal artery, renal vein, ureter, renal cortex, renal medulla, renal pelvis. So if you take one of the renal pyramids, you can see clearly the nephron part, where you can see Bowman's capsule, renal corpuscle, glomerulus, PCT, that is proximal convoluted tubule, DCT, distal convoluted tubule, lower part loop of Henle, collecting duct which moves towards the ureter. Then towards the left top you can see kidney, how the blood moves inside, and then the unwanted part coming outside. So you can see with the coloration red and blue, whereas lower part you can see what the picture shows, the renal pyramid, part of the kidney. When you highlight it, zoom through it, you can see various nephron present, and the nephron mainly the Bowman's capsule and the glomerulus. Okay. Then lastly the assignment question based on the today's part of the video. Thank you all of you.